physical evidence of the fossil record. Today we're going to basically kind of talk about the fossil record, and the video covers. Anybody watching that? So I should point out, just for your edification, it's a two-part series called First Life by David Attenborough. And so the video that's online is going to be the first part, which covers up through the Ediacaran, basically. And then the second video covers Cambrian explosion, arthropod diversification, invasion of land, things like that. I couldn't find a version of part 2 online. BBC's a pain in the butt about having things available online. Only the first part is online as a whole video. Part 2 has kind of been chopped up into little pieces because that way it's easier, I guess, not get recognized or something. So you could watch, or you could find it, catch it some other way. Watch part 2 of the thing is worthwhile doing as well, but the worksheet only covers the first episode. Anyway, so we started talking briefly about fossils. So fossils are basically the remains of dead organisms. And one thing I just wanted to kind of keep in mind when we're talking about the fossil record is that the record itself is heavily biased. It's biased towards animals with hard parts, with bones and shells. They're much more likely to leave fossils than soft, squishy things. It doesn't mean we don't have fossils of small, squishy things. It just means that those fossils are much, much rarer. And also, when we have just hard part fossils, it means we're missing a lot of the anatomy of the organism often. In addition, aquatic species are much more likely to leave fossils. That doesn't really matter so much for the early origins of animals, perhaps, but certainly later on, once animal life moves onto land, again, that's going to bias what kinds of fossils we see and what's more common. So the fossil record gives us a very incomplete picture. Only a very tiny percentage of the species that have existed have ever been discovered as fossils. All right. So a couple of things we want to know, let's see, here we go, first of all, how old the fossils are, and we can determine their age in 2 ways. One, we can determine kind of the sequence of which is older and younger, using what's called generally relative dating. That is, you figure out where in a stratigraphic sequence fossils occur. That is, sediments are laid down on top of each other, so older sediments are going to be at the bottom, younger sediments are going to be at the top, and so the relative sequence in any one location tells you which is older, which is younger, and so on. So you can build a sequence. However, if you want to connect different parts of the world and find if something living in North America is the same age as something in Europe, then you need to use indicator fossils, which basically can be used to correlate layers across different places. And so places with the same indicator fossil would basically be at the same age. These are generally often small, abundant marine fossils that evolve fairly rapidly, so they often have a fairly short window of time when they're in the fossil record and can be used to kind of date those particular sediments. So the relative dating tells you which is older, which is younger. It allows you to build sequences. And this is the oldest type of dating. It's been around for a couple hundred years. But in addition, we want to know how old things are in absolute terms as well. Absolute dating. We want to know how many millions of years that fossils date to. And to do that, we age rocks from their formation using radiometric dating, radioactive isotopes that undergo decay. We can look at we know the decay rate, which we know for all of these isotopes. That's the half-life. And so we know the half-life, the rate at which it decays. We can then look at the proportion of original isotope versus derived isotope, the daughter isotope, and use that to calculate the age. We're not going to get into any of the details here. But the one big limitation here is that radiometric dating, basically dates rocks from the point where they cooled off and no longer were lava or magma. So it determines the age of rocks from its volcanic origin. That means that most sedimentary rocks which are basically ground up sand and mud and so on, which are pieces of larger rocks that have eroded into sediments and then glued back together, cemented back together. So things like cement and asphalt, for example, are man-made sedimentary rocks. The problem is, if we try to date those, most of the time, there are certain exceptions when you have volcanic bits and pieces in there, but most of the time those sedimentary rocks just tell you the age of the parent material, the rock that it came from, not the age when the sediments were formed, and therefore not the age of the fossils in those sediments. And so we basically have to look for these volcanic layers to put upper and lower limits on particular air sediments. And sometimes those are we can get very precise dates, other times we don't get very precise dates. And there's a few cases where some kinds of bones and shells have been dated directly using isotopes, but that's fairly limited, mostly because it's only works with certain isotopes that are in decay relatively fast. So it only goes back 1 million years or 10 million years or something like that. Same with carbon dating, which is a similar idea. You're actually dating organic material directly, but carbon dating only goes back, is only useful for maybe about 60 or 70,000 years into the past. So not really useful for much of the life on Earth. Anyway, we put all of these, we put the combination of radioactive isotope dating and relative dating together, and we can kind of build a sequence of rocks, and using a fossil in there, we can kind of get an idea of the history of life on Earth. And so that allows us to build a geological timescale here. And this is a complete geological timescale. Basically, I'm not sure what a Priscoan is. That should be, well, never mind. Anyway, it goes back to the formation of the Earth 4.6 billion years ago, all the way up to the present. So this is basically the history of life on Earth. And what you can see is well, you can't really see it here because it's not labeled, but life originated about 3.8 billion years ago, probably. That's when we have the earliest evidence of life. We have some actual fossils that date back to about 3.5 billion years ago, some putative fossils that are less certain, about 3.8 billion years ago. There's some chemical evidence for biological processes going back about 3.8 billion years ago as well. And that's probably fairly shortly after the Earth actually got cool enough to support things like liquid oceans. So for much of this, life wouldn't have been able to exist anyway. So life appeared probably pretty quickly on Earth based on the limited fossil record. And I should point out that the older you get, the fewer rocks of that age are generally going to be available. That is, a geological cycle and geological processes. As rocks, as bits of continents subsume underneath the other, they get molten back, molten down. So old rocks are much more likely to have been destroyed by geological processes than young rocks. So the further back we go, the fewer and fewer rocks of suitable age we have. But anyway, so about 3.8 billion years ago, the first life, up to about maybe, let's say, 700 million years ago, all we had was basically simple organisms, bacteria, archaea, protozoan-like animals, simple algae, and that's about it. No animal life showed up in the fossil record, no definitive animal life showed up in a fossil record until somewhere around 700 million years ago. Probably actually appeared a little bit later, about maybe 800 million years ago. So that means for literally 3 billion years, life on Earth was basically a little more exotic than pond scum. And since this isn't a class in pond scum, we don't care about that. So we're going to kind of look at the end here. All right, and so you'll note, so notice that here's the Proterozoic, the Archaean. This is usually called the Hadean, I think, but the sort of pre-life period. Proterozoic began about 2.5 billion years ago. That's about when we see an increase, a permanent increase in oxygen presence on Earth. Much lower than today, maybe only about 2% oxygen, but 2% is better, much more significant than 0 before that. And up to about 540 million years ago, here is a Proterozoic, and then this is a Phanerozoic eon. I'm not sure why These are drawn over to the side, but anyway. All right, so the Proterozoic eon, which we're going to get in, which was where we start our story in, started 2.5 billion years ago, ended 542 million years ago. Remember, big M is a mega annum. That was replaced by the end of the Proterozoic eon, indicated the start of the Phanerozoic eon. The Phanerozoic eon began 542 million years ago, and we're still in the Phanerozoic now. It continues to the present. The Phanerozoic eon and then is subdivided into 3 eras. The Paleozoic from 542 to around 251. The latest dates I'm seeing actually suggest maybe more like 252 as we adjust our dates. The Mesozoic from 251 to about 65.5 million years ago. And then the Cenozoic from about 65.5 million years ago to the present. And so the transition, and so these divisions here are determined by geological evidence, by changes in the Earth. So the start of the Proterozoic eon, as I mentioned, is marked by a more or less permanent presence of oxygen in the atmosphere, much lower than today, but that means that there was extensive photosynthesis and things like that. The Phanerozoic eon is marked by the beginnings of sort of modern animal life. We'll come back to that in a little bit. And then the eras are subdivided. For example, the boundary between a Paleozoic and a Mesozoic is marked by a big change in the kinds of organisms that were present, marked by a mass extinction at the end of the Paleozoic. Same with the Mesozoic, a mass extinction there changed the kinds of organisms that were present. So all of these were basically, these boundaries are marked by changes in the fossil. Most of these eras and so on, and the periods, which are subdivisions of the eras, were identified and named and so on long before we were able to put dates on it. So you could just use the fossil record, relative dating, and kind of categorize the different time periods based on the dominant types of fossils. And then subsequently, in the last 70 years or so, we've been able to start putting actual dates to these things. So each of these eras is divided up into periods here. The ones that are probably most important for what we're going to be talking about now is going to be the Ediacaran, which isn't labeled on this particular diagram, but belongs here. It's the last of the Proterozoic in the Proterozoic. And then the Cambrian, which is the first era in first period within the Paleozoic era. Okay. All right. And as I mentioned, a change in eras and sometimes the changes in periods is associated with a changeover in the types of organisms that were present, and that in some cases indicated the presence of a mass extinction. And so there were 5 of these big sort of mass extinctions where the overall extinction rate was much, much higher than the sort of normal background extinctions. The 2 biggest were the one at the end of the Paleozoic era, so that's 251 million years ago. About 95% of all marine species went extinct. On land, it was a little bit better, maybe about 80–85%, including the last of the trilobites, all of the Paleozoic coral communities, and many, many other groups kind of went extinct at that point as well. Then the one that is probably most familiar is the one that occurred at the end of the Mesozoic. That would be 65.5 million years ago. That saw the extinction of the ammonites, a group of shelled mollusks. You meet eventually in lab. The end of the dinosaurs, marine reptiles, pterosaurs, and so on, all went extinct there as well. And at least in some cases, we don't really fully understand what caused these mass extinctions. In some cases, we have a decent idea. For the Paleozoic extinction, this big one, the end Permian extinction, the cause there is probably, again, there's less, the evidence isn't quite as strong, but it's getting better, that probably it was largely triggered by massive volcanic eruptions. And we're talking about volcanic, not, like wimpy little volcanic eruptions like the supposed, Yellowstone supervolcano, which would be wimpy in comparison. We're talking about volcanic eruptions that could bury literally half of North America under a mile of lava. We're talking about massive volcanic eruptions that are coincident with this. Those volcanic eruptions probably would have produced on a short term there's some evidence for short-term cooling due to blocking the sunlight and so on. That was probably relatively short-lived, maybe a few years. But following that, a lot of the gases that were released by volcanoes include CO2 and other greenhouse gases. So there's actually an episode of global warming that followed that mass extinction with the average temperatures going up 6 degrees Celsius or more. And that is considered probably the main cause for a lot of these extinctions following it. The one at the end of the Mesozoic is actually the most recent of the mass extinctions, not counting the one we're perpetrating right now. And is also, because it's the most recent, also the best understood. And in this case, the evidence is very, very strong that the extinction was caused by a massive meteor impact that hit the Yucatan Peninsula off of Mexico and basically caused an impact winter effect. That is, first you have the sort of blast, the shock wave and heat wave kind of circling the globe. This is followed by massive amounts of debris being put up into the atmosphere and darkening the skies. Hot molten rock coming back down would have started global forest fires, adding more soot and smoke, basically blocking photosynthesis for anywhere from 8 months to a couple of years or so, depending on the models involved. And that basically caused severe disruptions in food chains and ultimately a mass extinction. Okay. All right, so let's go back to animals. As I think I already mentioned last time, molecular clock data suggests that animals diverged from their protozoan ancestors, the Joanna-flagellates, somewhere around 800 million years ago. Yeah?
Do you have to memorize all the years that everything happened?
You should know some of the biggies. You should probably know you don't have to know the individual periods, except know the date of the Cambrian. But certainly, because we're going to spend some more time on it, this one, 542 million years at the start of the Cambrian is kind of an important one. The start of the Ediacaran, which is about 640 million years ago, 630 something million years ago. Those are kind of important. It gives you a little bit of a perspective of how they fit in. All right. The 800 million year one I'm not really that concerned about because that's kind of a rough data anyway, or this one here, except maybe just that sponges are a little bit older than even the first Ediacaran life. All right, so one of the earliest fossils that have been suggested might be a sponge is this fossil. It's from Namibia in southern Africa. It's about 760 million years old. It's not very, this is a scanning electron micrograph. It's about 0.5 millimeter long, so barely visible with the naked eye, a little tiny grain. And it looks kind of like it might be spongy, because sponges being kind of irregular and asymmetric, anything can kind of look like them, I suppose. So this has been suggested to be a sponge, but hasn't convinced everybody, but it's about 760 million years old. There's some chemical evidence for sponges. So this is a field that's really just starting, but we're now getting to the point where we can actually take a little bit of fossil and subject to a fancy little mass spectrometry and all of that, and actually identify chemical traces of the organism that actually made that fossil. So some of its molecules would have bound with the minerals present by the time it had fossilized, and some of those chemical traces can be still identified today. And there's some chemical evidence for sponges basically dating back to about 700 million years ago. And again, that's not particularly surprising. that you would expect the simplest animals, probably the earliest animals, to appear before anything else. So those dates aren't really particularly out of line. It's likely during that period there were some basic sponges around. But things kind of start taking off about 635 million years ago, which is when we start seeing complex animals and other weird things in the fossil record. We start seeing fairly complex life in the fossil record about 635 million years ago. And that brings us to the Ediacaran, or I have always been saying Ediacaran, but I think it's actually technically pronounced Ediacaran. So let's see if I can get in the habit of getting it right. The Ediacaran period dates from 635 million years ago to 542 million years ago. It's the last period of the Proterozoic. And we covered this already in the lab a little bit. Ediacaran animals were generally small, they were generally soft-bodied, so that means they don't fossilize well, which means that fossils of them are fairly rare and not always particularly well preserved. There were some hard parts, including the presence of sponge spicules, so we know sponges were present. There are some little things that look like they might have been worm jaws. And there are these guys here. This is a drawing of what a fossil called Cloudina, which had these hard skeletons that look like a series of sort of concentric cups standing on top of each other. And these actually formed early reefs, sort of hard structures built by animals. We don't know what kind of animal it was. The suggestions are that it might be cnidarians, something coral-like basically, or possibly some sort of secreted worm tubes. So it's kind of a little bit of anybody's guess. And these are from the fairly late Ediacaran. So mostly during the Ediacaran period, animals did not have hard parts. There weren't very many fossils. All right. So what triggered the rise of the animals during the Ediacaran? Well, the start of the Ediacaran is also basically coincides with the end of the Cryogenian period. The Cryogenian is named because that was a global ice age. Literally the entire Earth was frozen over with glaciers extending all the way down to the equator that had lasted for, I think, estimates suggest about 100 million years. So we had this global ice age where not much was going on, and that ice age basically eventually came to an end, presumably as CO2 increased in the atmosphere from things like volcanoes. That caused CO2 is a greenhouse gas. It can be helpful in some cases like this. That caused global temperatures to increase. And once you get warmer temperatures, you can have life blossoming, using up built-up nutrients and things like that. At the same time, towards the beginning of the Ediacaran was also marked by an increase in atmospheric oxygen levels. And we're not really high compared to today. Today it's about 22%. I think back then it would have been, going up to about 10% or so. And the origin of nitrogen-fixing bacteria, cyanobacteria, that can capture N2 from the atmosphere and convert it into useful forms, and thus makes that nutrient much more readily available to algae and subsequently animals as well. So all three of these are kind of associated with the start of the Ediacaran and may have helped give rise to more diverse animal life, some of which can be shown here, and some of which I'll talk about here in a second. So, all right. So, what did some of these critters look like? Well, probably the most common types of fossils from the Ediacaran are animals which we now classify as belonging into a clade called the Bendobionta. Sometimes you'll also see the term Rangiomorpha. I think that they actually just reflect different levels in the taxonomy, but not entirely sure. We're just going to call them the Bendobionta. So the Bendobionta are these these attached structures mostly. Some may lie flat, but many of them are attached to a substrate. And then they have this kind of frond-like structure with a very distinctive quilted pattern, and not like any sort of organism alive today. When they were first discovered, their shape is very similar to something like sea pens that you saw in lab. And so people thought maybe there was some kind of cnidarian, but they don't have any polyps or anything like a sea pen does. Their actual structure, when we have good fossils of them, indicate that they're fairly quite distinctively different. There's no evidence of a mouth or digestive system or anything like that. And so they may have absorbed nutrients directly from the water. We don't actually even know that these are animals. So there could be some sort of other protost offshoot for all we know for sure. They may be related to another group which we do not have evidence that they're animals, so it's possible they're animals. We don't think they were photosynthetic because some of them are found in fossil beds that represent relatively deep waters where there wouldn't have been any sunlight. So they could grow in deep water with no sunlight. So they weren't photosynthetic. They weren't just seaweeds. But how they made their living, our best guess is probably just absorbing dissolved nutrients from the water. They don't seem to have much ability to do anything else. Anatomy. This is a Eternia here. Well, actually, it's unidentified. I think that shouldn't be there. This just shows a painting showing one of these in nature. I put this in just to kind of remind me that. So a recent study actually looked at how these are arranged and suggests the reason for them growing tall is probably not feeding. There isn't a kind of a staggered or layered kind of community, which you might expect if they're each making use of different resources at different water heights. And so the reason for growing tall is perhaps best for dispersing offspring. And so they might have used that to disperse their propagules, and because the higher you are, the further they can get carried before they settle down. In terms of their anatomy, it turns out that their body plan can be produced by some very simple fractal branching rules. So you basically start with an axis, you add a branch to the left and one to the right, and you subdivide, then each of these gets subdivided. And so you can build a simple mathematical model. That's what these are here. A simple mathematical model that just tells you when to branch, a basic fractal math. And those fractal math problems will actually produce shapes that actually resemble actual members of this group. So their growth appears to be triggered by some fairly simple fractal math rules, branching patterns that produces their frond-like growth in life. All right. So another group that might be related to them or might not is a group that's been named the Proarticulata. These are flat animals that seem to sit on the bottom, on the substrate, on the ground. And they have what's called pseudo-bilateral symmetry, or sometimes called sliding symmetry, where they appear to be more or less bilaterally symmetric, but they're not perfectly. You can see that the segments, called isomers, here are offset. They're not, those segment divisions don't go straight across, but they're offset from one another. So this one is offset by 50% from the next one across the way, and so on. So this is sort of pseudo-symmetry or sliding symmetry here, rather than the sort of typical kind of transverse symmetry we get in modern animals. What are these critters? Some have suggested they might be related to the Bendobionta. They have some at least superficial similarities. They might be flat versions of them. There's also some evidence, I think that's coming up, here, from two different pieces of evidence now that suggest they're actually animals. One is that looking at a whole, at least for Dickinsonia, which is one of the more common species of proarticulates, at least for Dickinsonia, the way they grow is more typical of animal types of growth rather than plant growth or algae growth. So the growth patterns is more consistent with it being an animal. And again, biochemistry associated with some of these fossils, particularly the presence of particular cholesteroids, which are indicative of animal tissues, also kind of point to these guys as most likely being animals. Where they fit into the phylogeny of animals, we don't really know. There's not enough, we have kind of fossils that give us the overall shape, but we don't know anything about their internal anatomy. We obviously have no molecular data or anything like that. So where they fit in terms of animals is not known at all. All right, another group, so there's a bunch of round fossil, sort of more or less circular fossils from the Ediacaran fauna. Some of those circular forms were basically holdfast, the attachment points of these stalked animals. But there's at least one group called the Trilobozoa, which have a distinctive threefold symmetry. So you can see here there's kind of three main arc-like ridges. On this top one here, you can see there's kind of a threefold symmetry here. Some of them have small canals, like the radial canals on the jellyfish. So the suggestion is that these are probably related to cnidarians. We don't know for sure, because they're not terribly similar to any modern cnidarian group, but that seems to be sort of the best guess, that they were probably some kind of an early cnidarian offshoot. They were probably suspension feeders, and we can tell that based on some flow modeling. That is, you take these, you build a three-dimensional model of one of these guys in a computer, and you model water flow from various directions, and you see where the eddies and currents kind of form and take the water to. And it tends to, the flow around these guys tends to be consistent with bringing food into feeding areas. So they probably use their shape to basically draw currents into them and trap small food particles. It's kind of our best guess. All right. The basic point here with all of these guys is that most of these critters don't seem to have any really close relationship to modern animals. It's possible these guys are related to Cnidaria, but they're quite distinct from any modern Cnidarian group, so that's not really an absolute certainty. And the Proarticulata and the Vendobionta, they're just so weird, it's, again, arguable whether they're even animals. Whoops, and I just did this button twice, sorry. There are some fossils that do suggest a relationship to modern animals, so we're starting to see phyline. So here are some examples. So this is a reconstruction of a fossil of an animal called Hautia, something like that, which has been interpreted as a possible Cnidarian polyp. There's been fossils that are interpreted as a Cnidarian medusae. There's a fossil that's interpreted as a possible soft-bodied trilobite, sort of an early arthropod, but without the hard shell. Kimberella, the guy up here, has been interpreted as a possible mollusk because of a mollusk-like feeding structure. And Parvancorina here has been, again, interpreted as a possible early arthropod or arthropod relative, whose shape also suggests it was probably a suspension feeder. So there are some critters amongst the Ediacaran fauna that suggest at least the early hints of modern animal groups. And we also have trace fossils as well that suggest that animals were moving around. So there are, for example, trackways. These are trackways that are 585 million years old. And so that means that something was moving around using muscles, including that may be worm-like animals that would have been bilaterally symmetric, or maybe a anemone-like animal which can move along substrate sometimes. And even the presence of a slightly more recent 551 million year old fossil indicating or several fossils indicating a presence of paired appendages. You can see there's individual footprints here, and there are two double row indicating paired appendages or legs. That basically tells us it's an arthropod of some sort that was crawling around 551 million years ago. So we can argue that at least some major animal groups started to differentiate in Ediacaran, even though most of them were probably soft-bodied. So our ability to find fossils of them is very limited. And so just some pictures of what these Ediacaran environments might have looked like. So you had these Vendobionta, these sort of frond-like things here, various sizes and shapes. There's quite a bit of variety of them. You obviously would have had jellyfish-like critters. You would have had these flat proarticulates here. Some of them might have even been swimming around. Here's one of those trilobozoans. Let's see, I've got a few more pictures here. I think this is from this, diorama at the Smithsonian, showing again some of these Charnia type vendobionts, some Dickinsonia type things swimming around, some jellyfishes, etc., and some algae in the front as well. Here's just another picture showing there's Trilobozoa, Parvocarina. This is probably Dickinsonia, Sperdina, Charnia, and some whatever that thing is up here. Probably some early sponges here. So this is what life might have looked like back around 550 some odd million years ago in the ocean. All right. So next comes the Cambrian period. So the Cambrian period marks the beginning of the Phanerozoic eon. It's the first period within the Phanerozoic and therefore the end of the Ediacaran period. So the Cambrian began 542 million years ago and ended 495 million years ago. I don't really care about the ending date, since I know you're going to ask. But that 542 million is pretty important. It's the start of the Cambrian, it's the start of the Phanerozoic eon, the end of the Ediacaran, and so on. So that's a fairly big date there. Anyway, what marks why we actually mark why this is the transition from the Paleozoic, why this marks the beginning of the Phanerozoic eon is because this is when we start to see modern animal phyla in abundance, and we see the appearance of animals with hard parts, with shells, with spines, with exoskeletons, and so on. And the appearance is pretty quick. That is, at the very beginning of the Cambrian, we start to see the accumulation of the small shelly fossils, small animals with shells, little spines, little like bivalve shells and snail shells and tubular shells and so on, mostly very tiny little guys. And within about 20 million years, we get a much more diverse Cambrian fauna with much larger animals, bigger shelly things, and so on. So that first 20 million years started off with just small shelly things, leading to the sort of more typical, what we think of as the typical Cambrian fauna. And during that Cambrian period, we see the appearance of pretty much all of the major animal phyla that are still with us today, as well as a few weird groups that might not represent any modern phylum. And so this is over from a geological perspective, a fairly short period of time where we see this sort of sudden rapid diversification of animal life. And so it's been referred to as the Cambrian explosion, so big boom of animal life. All right. So we know about this from there are Cambrian fossils around the world. But the best sites are sites that preserve not just hard parts like shells, but also soft-bodied organisms as well. Because even though we see the appearance of many shelled animals, there's still a lot of soft-bodied critters around as well. And two sites in particular are especially important. The Burgess Shale in British Columbia, Canada, which dates to about 505 million years ago, about the Middle Cambrian, and the Chenjiang biota in China, which is a more recently discovered site, dating to about 520 million years ago. So it's a little bit older. But there's a fair bit of overlap in the kinds of organisms they have that occur at both sites. Both of these are known as Lagerstätte, which is a German word that's usually roughly, not quite literally translated as motherlode, but it's basically used to identify sites with exceptionally good, exceptionally exquisite fossil preservation, including the preservation of soft-bodied animals, not just hard parts. And so that's where there's some other Cambrian sites as well that are pretty decent preservation, and lots that preserve at least the hard part kinds of organisms. And so so we see most modern phyla, but we also see some bizarre groups that we don't necessarily know how they fit in with modern phyla. So I'm just going to give you a bunch of pictures here, so because pictures are pretty nice. And so these are just some examples of some of the Cambrian animals. So here we have Fujianhuia, which is an early arthropod, an animal with jointed legs. Wubaxia is generally thought to be a mollusk, a very primitive basal mollusk. Odontogryphus is also a very early mollusk, at least probably a mollusk. Otoya is actually a pretty well-known species. Is a preopulid worm, a group that worms that still exist today, although they're not very species-rich. There's like 12 surviving species. They were actually more alive during the Cambrian than there are now of this group. We have some other weird exotic animals include hyolithids like this Haplofrentis, which was actually up until fairly recently. Nobody really knew what they were, but a study of literally thousands of fossils found some that preserved a ring of tentacles called a lophophore, and that and a combination of shells, a microshell structure, suggests that these are related to a modern group called brachiopods, which you will see in lab on Friday. So keep this guy in mind. We'll see him soon. Others include possible onychophorans. Modern onychophorans are called velvet worms and are found in tropical rainforests. They're terrestrial, but they were marine relatives of these guys living back in the day, including an animal called hallucinogenia because it was so weird. And the original discovery was reconstructed upside down on top, so it was thought to walk on these spines and had a row of sort of like tentacles on its back, but those tentacles turned out to be paired legs. And so it's still pretty weird looking, but maybe not as weird as it was when it was originally discovered. There's another critter called the walking cactus, Diania cactiformis. That's from China. That also may be related to these guys as well. And you guys had models of these when we did our Cambrian lab, and some of these you'll see again when we got to the arthropod relatives lab. I'll bring these models out again. We've got a model of this one you'll see next week, this week, sorry. To show some of the worms during the Cambrian. There's actually quite a lot of worm fossils, and so including, I think this is probably Otoya here, and some of its periopulid relatives, but also segmented annelid worms, leaving fossils as well. And then we have various Cambrian arthropods. So the arthropods are the biggest modern phylum, the animals, insects, crabs, spiders, all that good stuff. And there were quite a diverse array of them in the Cambrian. So remember, we saw some possible arthropods in the Ediacaran, but by the Cambrian, we got lots of them, including Canidaspis, which has a big sort of shell here, much like modern day Daphnia would have. Morellas, these sort of lace crabs, although they're not actually true crabs. Yohoya, another weird little early arthropod. Olenoides is a trilobite, so you see the first trilobites, which became a very successful arthropod group. You also have some arthropod relatives, including Anomalocaris, which was the largest predator to have ever existed up until that point on Earth. They could get up to about 1 meter long, and had a round mouth full of sort of jagged teeth plates that basically acted as teeth, appendages that it could use to grab food, big eyes, and some of its relatives were filter feeders or mud sorters and so on. Another weird group that's probably related to these guys is Opapinia, which has 5 eyes on the ends of stalks, and a long proboscis with a sort of grabber at the end. And then the mouth was actually here, so the mouth was not at the end of the proboscis. And you had other members of modern groups, so you had things like, this is an early annelid worm from the Cambrian. This is Metaspergina, an early chordate, a relative of our own phylum, or part of our own phylum. This thing up here, it might not look good. This is Tino imbricata. It's actually an early echinoderm, member of sea stars and sea urchins and things like that. But unlike modern sea stars and sea urchins, which are basically radially symmetric, this represented an early bilaterally symmetric ancestor when they were still bilaterally symmetric. And then things we just don't really know what the hell they are. So there is this herpetogaster, which might be something related to echinoderms, possibly, maybe. You have orthoxanthus, which we don't really know what it is. And Amisquia, again, is not really known what it is, although its overall shape is superficially a little bit like arrowworms. So that might be a link, but they're they're quite a bit bigger, proportions are off, and we're not really sure. So these are just some oddballs that have been known from the Cambrian. And where are we on time? We're out of time. I think I probably don't even want to try the next slide. Well, yeah, we'll stop here because now we're out of time. We'll go through some pictures and a little video clip, doing more Cambrian stuff. And